He will talk about the patient-tailored medicine, the new trends in healthcare. Please.
Thank you for the kind introduction, and thank you to the organizing committee for inviting me to come to this amazing meeting that you're hosting. And congratulations to SCL for your history, putting on this great event, and really look forward to what you're going to achieve in the next 40 years. So thank you for inviting me to be part of this. Today, I'm going to talk about some aspects of precision medicine as it pertains to cancer care, where I think there's a lot of advances that are currently happening. I'm going to make one of the disclosures. I founded Protean Biodiagnostics 7 years ago. Protean is a company that's trying to close the gaps in American healthcare to bring precision oncology to more patients across the United States and across the world. We are fortunate to be living in a really revolutionary and exciting time in medicine. We are moving from traditional medicine in the care of cancer patients, which was reliant on surgery, chemotherapy, and radiation therapy, to a new era of precision oncology, where we can really adapt and target therapies specifically for patients and avoid toxic side effects and get maximum effect. Basically, precision medicine, as we know, is the correct diagnosis, the correct time, and the correct treatment. Now, one thing we know about cancer is a tricky disease. It changes from the time of diagnosis, it changes when you treat it, and it develops different mechanisms of resistance. So a patient's journey with cancer is truly a journey. It starts perhaps even at the time of their birth with their genetic makeup, with their exposure during life, and with their cancer. And the treatments will also alter the cancer and cause different alterations. So being able to capture this and measure this is important for selecting appropriate treatments. If we consider random selection of patients, none of our therapies would be approved because their response would be very small. However, if we use a companion diagnostic and pre-select patients who are likely to respond, we're actually going to see much more effective response to particular therapies. Hence, the future of precision medicine is really pre-selecting patients for appropriate treatment. Now, this is not as easy as it might sound. One of the biggest challenges facing precision medicine is actually the explosion of knowledge. If we look at these charts, the chart on the left, the blue line, indicates the number of FDA approvals of new cancer drugs each year. Back when I was a kid in residency, we'd only get 1 or 2 new cancer drugs every year or two. There weren't very many. Last year I checked, there was over 200 approvals, almost more than 1 a day for new cancer drugs. This is tremendous. However, many of these cancer drugs also come with companion diagnostics. And this puts challenge on the lab system. How are we going to provide all of these cancer companion diagnostics that are all quite different? The middle chart shows the biomarkers and the growth in potential cancer biomarkers year over year. Phenomenal growth from the research. Now, the network at the far right, no, it's not the nodal network of the United States. It's actually the treatment options for cancer patients. Each red dot is a cancer. Each green dot is an approved therapy. There's dozens of therapies for each cancer, and there's more and more occurring. How is a doctor to manage this? How can a doctor cope with this incredible explosion of information? A recent survey by the American Society of Clinical Oncologists indicated cancer disparities and disparity in treatment is one of the biggest problems facing American cancer patients and cancer treatment globally, in that the advances are outpacing our ability to deliver them, and more patients are falling behind. This is a huge problem. In fact, in the United States, over 85% of patients are treated outside of academic medical centers where they get the best possible care. So these are being treated in community centers. A recent survey showed that over 60% of oncologists, now these are the ones that admit they don't really know what's going on and how to manage all of this information. A recent study showed that over 30% of cancer patients receive incorrect diagnoses and incomplete diagnosis. That's shocking. If you don't have the right diagnosis, how are you going to treat the disease? Less than 5% of cancer patients get onto clinical trials. This is abysmal. We really need to get more cancer patients onto clinical trials, and also underserved populations, particularly in the United States. African Americans, Latinos, and others are drastically underrepresented in clinical trials, and this is a huge problem. And finally, over 50% of lung cancer patients currently do not get proper molecular testing and treatment, despite these being on the guidelines for more than 15 years. In fact, a recent study that was published last year at ASCO, done by DeciBio, showed that only 35% of American lung cancer patients actually got EGFR testing and treatment. Only 35%. And this was done by a survey of over 50,000 Medicare records. So they identified over 7 pitfalls, 7 gaps where patients are being lost. And this is a huge problem. If we can't treat patients with EGFR, how are we going to treat all of the other ones? Now, the treatment of lung cancer is really a poster child for precision oncology. When I started as a pathologist about 30 years ago, we only cared, was it small cell cancer? Was it squamous cancer? That's all we really had to report. Whereas now there are dozens of molecular classifications for lung cancer, and each one of them have a specific therapy. So it's imperative that we actually perform this testing and make it available to physicians and patients. Not forgetting our work as pathologists, immunohistochemistry is interestingly coming back into the limelight again. There are more treatments that are targeted at proteins. The grandmother of all biomarkers, in my opinion, is estrogen receptor, that it's been a target for breast cancer treatment for over 50 years. And our ability to measure estrogen receptor is somewhat lacking, in my opinion. Somewhat it's subjective. A lot of us are still using an Alred system where we estimate the number of positive cells and hope that tumors that have more than a certain number will respond to treatment. Now, this led to some problems in Canada, where I practiced for many years. It turned out that one of the Canadian provinces misaligned or miscalibrated their estrogen receptor test. And this province continued to use an inappropriate control for over 10 years and affected hundreds and hundreds of patients with misclassification of ER positivity and negativity, giving rise to problematic treatments. And this is in a Canadian system with a central lab that ran into this problem. This set up a national program in Canada where we tried to compare the labs, and this really shows the results. Each column is a different lab, and each row is a test case. So we've put them through the different labs and said, oh, this is pretty good. There's about a 90% concordance. That's great. Is that great? So if you take a patient up there, some of the patients are categorized as being negative in some labs, in other labs it's positive. That would define 10 years of endocrine therapy on the flip of a coin. I think we can do better. This is problematic, in my opinion. Another marker that's really become important in cancer therapy is PD-L1. Now, PD-L1 is a key marker for selection of checkpoint inhibitors, and I think this marker is a particularly good marker to understand how not to set up a companion diagnostic. With PD-L1, there's actually multiple clones available. So there's different clones that more or less stain the same thing. However, one of them seems to be out of alignment. That's SP142 that was developed by Roche. Again, the concentrations were slightly different, and if they reformulated, the mixture would come up to look very similar to the other ones. But this creates a problem in that there's 4 different clones or more for measuring the same target. That creates problems for pathology labs. Now, if we look at how to score this, it's like, are you kidding me? These are the scoring systems for PD-L1. There's multiple ones. There's 5 of them here. They were used in different clinical trials and have different cut points. You have to look at how many tumor cells are positive, how many immune cells are positive, are the immune cells inside the tumor or outside the tumor, and so on. Extremely complicated analysis, very hard to teach pathologists how to do this accurately. When humans are involved, it's problematic. Now, there is some help here coming along with in terms of digital pathology, in terms of digital analysis might be able to help us. There are some new technologies that allow us to stain in multifluorescence, and multifluorescence staining is superior to immunohistochemistry and DAB. Now, for those of you who are technically minded, the DAB signal is not linear. It's designed to be sigmoidal. It was really designed to define positives and negatives, whereas if you go to a fluorescent pattern, you can actually get a more linear signal, and it's a positive signal because you can adjust the exposure on a camera and collect the signal over time on photons. But it also allows you to stain with more than one stain at a time, so you can stain with multiple stains. So in this case, we can look at multiple stains that are available here to stain for PD-L1, tumor cells, and immune cells. Now what this does is allow us to analyze tissue and have the computer count the cells and calculate the various scores, the IC score, the tumor proportion score, and so on. And so we did a little experiment with this to look at how are these different scores correlated, and it turns out they're actually pretty well correlated in many ways. TPS is highly correlated with CPS, TPS is correlated with IC, and so on. And it seemed that there are really two clusters there, really strongly positive and negative. But the point here is that many of these scoring systems are somewhat equivalent, but we seem to have overcomplicated them in many ways. Now, the other thing to remember is that there are different approvals for these PD-L1 drugs, and also some of these drugs target different targets. So some of them target PD-1, and other ones are PD-L1. This may account for the different performance in different clinical trials, but it certainly complicates the picture for pathologists who are really trying to understand how best to analyze tissue for immune therapy selection. Now, if that's not bad enough, we're only at the beginning of immune therapy, and PD-L1 and PD-1 are only one marker that is a target on T cells. There's actually dozens of other targets that are available for pharmaceuticals to target, to both activate and inactivate T cells. So this problem is going to become worse in the future. There are other types of therapies that are emerging as well for immune therapy. These include types of cell therapy, even administrating natural killer cells, living therapies, different types of engineered T cells, viruses, and so on. So we're living in a very exciting time when it comes to therapeutic options for patients. However, it's very much a challenging time when it comes to pathology and how pathology can support all of these advances. I'm a personal fan of multiplex immunohistochemistry, and I've used it for over 20 years. And one of the advantages of multiplex immunohistochemistry is that you can really study the microenvironment of a tissue in a single section. So here's one section of a piece of tumor that's been stained with keratin, CD8, PD-L1, PD-1, CD3, FOXP3, and DAPI. By doing this in a multiplex stain, you can now isolate each of the components and look at the immune cells in the tissue microenvironment. You can preserve their arrangement and determine which cells are next to each other and which cells are inside of the tumor as well. So this technology, I think, we should be embraced by pathologists because it really can improve our ability to analyze tissue and really generate phenomenal data from the tissue microenvironment. Another area where there's a lot of advances is in AI and digital image analysis. Interestingly, a lot of the AI is focusing on H&E images that hasn't yet gone into the multiplex, but the H&E can be quite powerful. And at Protean, we've actually become quite interested in a problem called Barrett's esophagus. A large percentage of people develop Barrett's esophagus, and a small percentage of those go on to get cancer of the esophagus and stomach. We've been working to teach AI systems to classify Barrett's esophagus into ones that are predicted to advance and ones that are stable. And we've made some success in doing this, that we can reliably identify cases with Barrett's that are more likely to progress. So I think we'll see more of this in the future. When we turn back to cancer, it's again a very exciting time. In the last couple of years, particularly in some tumors, for example, cholangiocarcinoma, multiple therapeutic targets have now emerged in dozens of different genes. Now, what this means is the path labs need to be able to measure these in a consistent and reliable way in terms of looking at the various types of somatic mutations that are present. This has led to the development of large multi-gene panels. And there are several that are currently in production. Illumina has something called TSO 500. That's an adaptation from TST 170. It's getting broad global use. I quite like this panel for those of you that are looking for large panels. It covers over 600 genes. It also has RNA in it as well. So using a broad panel like this can help you categorize multiple types of cancer and identify therapeutic targets. You can also set it up in your lab to make the workflow easy, in that you can use the same panel for a variety of different types of cancer and really focus it down on what you need for particular subtypes of cancer. Another side effect of using a panel like this, a broad panel, is we can look at tumor mutational burden. And this by itself is an indicator of immune therapy response. We know that tumors may have different mutational loads, and on this chart here from the left to the right, we're seeing tumors on the left with low mutational loads and those on the right with high mutational loads. The highest mutational loads are carcinogen-induced tumors like melanoma due to sunlight exposure. Gives you very, very high mutational burdens. And these tumors with high mutational burdens respond well to immunotherapy. And it's thought that it's because they create neoantigens. So very interesting thing. Now, one thing when you're setting up assays in your lab looking at DNA and RNA, DNA is pretty easy to look at and is quite stable. RNA is also very important, and a lot of assays don't include RNA. I think RNA is important for a variety of reasons. One, it really tells you what's being expressed in the tumor and is the mutation being expressed, but you can also identify novel splice forms that can be important in identifying therapeutic targets. So when you're setting up an assay, you should consider also adding in RNA, because it can be quite important for your assay. Now, there's some interesting emerging technologies that I find fascinating. Here's one I just wanted to share with you. This is BioNano's optical genomic mapping. And what you're actually seeing visualized here are individual strands of DNA that are very long and that they're labeled with landmark targets on them. What it allows you to do is to isolate the DNA, label it, and do a de novo construction of the entire genome to identify areas of alteration. So by doing this, you can find fusions that might be missed, areas of chemotrypsis or chromotripsis, or other types of alterations. These two panels are the same patient. One patient, tumor 6 is their primary tumor, and ascites 6 is the same patient, the ascites fluid from the primary tumor. And you can compare the structural alterations that are occurring in the evolution of a tumor looking at these types of alterations. Now, the life of a pathologist. So this is what I get sent to me to analyze. What kind of tumor is this? I was like, well, what is this? Like it looks like a little bit of burnt dust. I don't know what it is. And often specimens come to me that other labs have used them all up and there's nothing really left. So this becomes a challenge. What are we to do? Well, not all is lost, because we now have liquid biopsy, and I think liquid biopsy is really transforming care for cancer patients. This allows us, with a simple blood test, to really determine what is going on in a cancer patient. Do they have residual disease? Is the disease progressing? Is it responding? Is it changing? What's going on with the disease? So tumor continuously leaks components into the blood, whole tumor cells, exosomes, pieces of DNA. All of these can be isolated, and it's really phenomenal to think in that tube of blood, you've got that whole patient's history. You got their genetics, you got what they had for breakfast, you got their blood type, you know what kind of viruses they've been exposed to, but you've also got all of these little fragments of their cancer in terms of the DNA, the mutations, and so on. So very exciting capabilities. And with this type of technology, we can use it for screening. So there's some tests that are coming up, for example, the GRAIL test, a pan-tumor test, that might be able to identify patients who have early stage cancer in a more accurate way than scanning. And it could be very useful. It can also tell us molecular staging. Did we actually completely remove the tumor? Or is there any tumor left behind? We can determine if the tumor is changing and if it's responding to therapy. So liquid biopsy is a very important technology, and I think we're going to see more and more and more of it as years go by. And there's a dozen different kinds of things that we can measure in the blood. I won't go over all of them for the sake of time, but obviously many, many things that you can measure in the blood. And some of them can be measured in very small amounts of blood. However, others you do need the volume. So it really depends if your particular marker, what the concentration is in the blood. Obviously, if you've only got 1 or 2 chances to get it in a mil, you're going to need a lot. But if there's millions per mil, you might be able to detect it in a small drop of blood as well. Moving quickly to genetics, I won't belabor this as there's some discussion earlier, but we understand that genetics is more and more important in cancer treatment, not only for risk of developing cancer, but if you have cancer, knowing your genetics can guide therapy for PARP inhibitors and for checkpoint inhibitors as well. So genetics is quite important, and now it's recommended every patient with ovarian cancer actually have a genetic test. It's starting to become more important for prostate, for pancreas, and other tumors as well. Now, this really highlights where we at with cancer care today. I think there's a large gap, particularly in the United States, where there are many advances, but they're not being deployed, they're not being implemented, and this is a big problem. This is one of the problems that we're trying to address at Protean, and to address this problem, we've created a digital health platform called MAPS. MAPS is a system that's cloud-based, that's integrated at Protean, that attempts to make precision medicine easy by helping the doctor pick the right tests, arrange the tests for them, and organize the data. So it's a one-stop system where the doctor doesn't even have to know what test to order. They're guided by national guidelines, by insurer guidelines. The tests can be arranged, they're ordered, they're provided to a doctor via a portal, and they can be arranged remotely. In addition, all of the data is available in the Azure cloud, a health secure cloud. It's HL7 compliant and FHIR compliant, so it can be interoperable with other health systems. We've endeavored to create a one-stop solution where you have the pathology, the molecular analysis, liquid biopsy, and genetics all in one place. It's dynamic, it's continuously updated, it's easy to use, it's available for doctors and patients to be able to use that through electronic portals and so on. We've also tried to make the report simple, so we integrate all of the information, including genetics, pathology information, other information, into a simplified report that even an oncologist can understand. The other thing that we can provide is tumor boards. For those of you working at academic medical centers, the tumor board is the most important activity where patients are reviewed, their case is discussed, and the team is there. In America, where patients are being treated at community centers, they don't have tumor boards. They're being treated by one-off oncologists or surgeons. This is creating a problem. So what we've done is we've partnered with Roche to create a virtual tumor board where actually doctors can participate in a tumor board that's virtual, where there can be other members or not. So within this environment, all of the patient's information is loaded. We've actually got this arranged that can be populated automatically from our health cloud system, and that doctors can easily review the information. They can consult with their colleagues, and within this tumor board tool, there's also additional modules that include guidelines, includes clinical trial matching, and other tools that can really help them pick the right treatment for their patient at the right time. It creates a portable record, so this can be shared if the patient is going to another center or if the patient is looking for eligibility for a trial. So making all of this data interoperable and integrated now allows the patient to be mobile through the system. The other component about the best practice of tumor boards is they should have multidisciplinary involvement that includes oncology, molecular, pathology, surgery, radiation oncology, and so on. And creating a virtual environment like this allows us to bring all of these experts together. We're using this quite successfully with patients around the US and even around the world. We have some patients in Australia and India that are using this particular product. And it's also helpful for their family members if they have family members that are scattered around to assist with consultations. Just as an aside, Protean also has a very active R&D component where we work with emerging biotechnology companies to find new diagnostic technology to rapidly implement into the system. The US system is complicated because there's several regulatory pathways. There's an LDT pathway, there's an FDA pathway, and so on. And at Protean, we work with many international labs and groups to help bring their technology into the United States. So if anyone is interested in working with us at Protean, we can work with you in partnerships and so on to bring technology forward. We also run clinical trials, and we're targeting underserved areas, so African American communities and various other types of communities in the United States to support clinical trials in those areas. Now, going towards finishing off, tissue management is the most important thing for cancer patients. And this is where pathologists have such a key role. The pathologists are the guardians of the tissue, that we are the ones that make a diagnosis of cancer. We're the ones that prioritize the testing and organize it, and we maintain these tissue blocks maybe forever. So they're incredible resources for both research and for the patient over the years. So pathology has such an important role in precision oncology and shaping the future of it that we really need to take our role in that very seriously. So in summary, we're living at a very exciting time in terms of precision oncology, and I really think that pathology is central to it. We've got amazing new technologies that are coming, testing requirements that only pathologists can help administer, and our role as pathologists are key to really be leaders in this area, to work with our clinical colleagues, regulators, and so on. Now, in the United States, there's terrible challenges that we are facing. The system is fragmented. The data doesn't move well. There's many choices of treatment, and there's inadequate specimens. So there's a lot of challenges that need to be addressed. There's delays in testing and so on. And one of the biggest problems in the US is the payment and regulatory systems, that the payment systems in America are very complicated with different types of payers, and really moving them forward so that we get proper payment for these types of services is key. And I think for that to happen, pathology really needs to play a key role in doing that. So I want to thank you for listening to my talk today. If you're interested, I also have a podcast where I talk about precision medicine as well. It's designed general, non-commercial for people who are interested, mostly patients, but others find it interesting as well. So I'd like to thank you for the opportunity to speak today, and I don't know if there's any time for questions, but thank you for that.
Thank you, Dr. Magliocco, and very